Inside almost every cell, there is many organelles, but most relevant to protein synthesis is the nucleus. The nucleus is a circular or oval-shaped organelle that contains DNA. DNA stands for deoxyribose nucleic acid, and it contains inherited information. DNA is made up of nucleotides, made up of a sugar molecule, a phosphate group, and a nitrogenous base. Each base is either thymine, cytosine, guanine, or adenine. Two strands of DNA are joined by weak hydrogen bonds and they are twisted into a double helix. These strands are ultimately just alternating chains of nucleotides, and they follow the complementary base pair rule. The adenine pairs with thymine and cytosine pairs with guanine. DNA contains thousands of genes. Genes are small sections of DNA that have a specific sequence of bases and are able to code for a specific sequence of amino acids, creating particular proteins. To actually create particular proteins, the sequence provided in genes has to be read by ribosomes, another type of organelle that is found outside of the nucleus. DNA is quite large and can't just leave the nucleus so that it can be copied by ribosomes. So, to make the protein, since we can't move the actual gene, we instead make a copy of the gene. This copy will be much smaller than the original DNA strand, meaning it will be able to leave the nucleus. But why? Why do we need protein synthesis? Protein synthesis is crucial for many cell processes. It's vital for the growth and repair of damaged body tissues, and proteins can also act as enzymes, catalyzing reactions that are necessary for metabolism and other cellular processes. So, these copied single genes are actually called mRNA, or messenger RNA. mRNA is quite similar to DNA, except for a few things. Firstly, mRNA is much shorter than DNA because it's only a gene long. Secondly, it's only a single strand instead of double-stranded like DNA. Thirdly, instead of the base thymine, mRNA contains uracil. To actually form mRNA, a process known as transcription occurs. So let's start by looking at the gene we want to copy. Before transcription can officially start, first, DNA helicase, a type of enzyme, unwinds and unzips the DNA strand. Then, transcription starts. This starts with an enzyme, a biological catalyst known as RNA polymerase. This enzyme binds the DNA a tiny bit before where the actual gene starts. As it binds, the hydrogen bonds form between the complementary bases, between the DNA and RNA, and then covalent bonds form between the adjacent sugar molecule and phosphates on the RNA. This causes the DNA strands to separate, so that all of the bases are exposed. At this point, the RNA polymerase continues to move along the unzipped and unwound DNA strand, reading each base so that it can be perfectly copied for the mRNA strand. This means that the mRNA bases will be complementary to the DNA bases, meaning that A on the mRNA strand will pair with T on the DNA. C on the mRNA strand will pair with G on the DNA. U on the mRNA strand will pair with A on the DNA. And finally, G on the mRNA strand will pair with C on the DNA. So, as the RNA polymerase reads through the bases, the mRNA produced will be complementary to the section of DNA being copied. This will continue along the whole gene, base by base. As this occurs, the DNA will simultaneously unzip slightly ahead of the RNA polymerase. This will continue into the end of the gene. At this point, the DNA also rezips and rewinds with the help of DNA helicase. It's also important to note that the DNA strand being copied to make the mRNA is actually referred to as the template strand. The side of DNA that was not copied is the coding strand. It's also important to note that in DNA and mRNA, each three bases can be referred to as either a triplet or a codon. These code for specific amino acids. Now, let's take a closer look at the mRNA produced, currently positioned in the nucleus of the cell. Because the mRNA is short enough to leave the nucleus via a nuclear pore, it can now attach to the ribosomes in the cytoplasm or attach to endoplasmic reticulum, meaning it's now able to undergo translation and eventually produce a protein. To start the process of translation, the mRNA strand and a ribosome both bind together. The ribosome begins to read the mRNA code, starting with the start codon and concluding only at the stop codon. At this point, tRNA, also known as transfer RNA, carries amino acids towards the ribosome. The tRNA is made up of amino acids at the top and an anticodon at the bottom. The codons on the mRNA strand and the anticodons on the tRNA attach by complementary base pairing. The second tRNA molecule attaches itself to the next codon on the mRNA in the same way. These two amino acids attached to the tRNA molecules are joined by a peptide bond through a process using ATP. This process continues as the third tRNA molecule binds to the next codon on the mRNA. This continues until reaching the stop codon, when the ribosome detaches and only the amino acid chain is left. At this point, the amino acid or polypeptide chain then bundles or rolls up, creating a protein. This means that both translation and protein synthesis